I'm Bahar, and I'm going to be talking about systematic reviews today. Systematic reviews is something I've spent the last 2 years kind of perfecting for myself. I've had to do systematic reviews as part of my PhD, and each one I do, I feel I'm just getting better and better at the technique I'm using and the strategy I'm using and the way I go about it. So I thought it would be great to share it with you. If you're interested in what a systematic review is, if you've done systematic reviews and you're just looking for advice on certain areas or certain steps of the systematic review to help things go more efficiently, to make sure you're doing things accurately, I hope I can answer most of your questions here. For those of you that don't know what a systematic review is, briefly, a systematic review is a systematic and scientific system or approach to collecting secondary data. Most of this data is from published literature that you can just find online. And the whole process includes a series of steps that you'll follow to collect your data. So step one is, what is your research question? What are you trying to find out? What answers are you looking for? This might be one of the easiest parts because you know why you want to do a systematic review. You're just trying to learn information about your topic, perhaps gather data to analyze, which is most of the reason why I do systematic reviews. So what is your research question? Secondly is creating your protocol. What are going to be the criteria that each paper, each study that you look at needs to meet the same inclusion criteria for your study? For example, inclusion criteria that I usually use is a time period. So I'm only interested in data from the year 1990 to present. So I will be excluding any papers before 1990. Another inclusion criteria that I use a lot is human samples. So I therefore exclude any papers done on animals, environmental samples, anything that doesn't come from a human, I'm excluding. So this is an example of different inclusion criteria that you will have. Another could be sample size. I like to limit my sample size to at least 10 samples or more. A sample below of 10 I don't find representative for what I'm looking at. So inclusion criteria. Next would be your search strategy. Your search strategy is going to consist of different terms that you can use to search these databases for the articles that you want. I've seen very simple search strategies, and then I've also seen really complicated ones. I do have to say, I think the more thought and effort you put into your search strategy, the better your systematic review will be. For the purpose of this video, I'm going to pick an example topic. So my example topic is going to be, do dogs like the color blue? I'm going to search through all relevant articles about dogs and studies on what colors they like to try and collect this information. So I'm beginning with my search strategy. Say I just make a search strategy of dogs, blue, and I put that into PubMed. I could be missing an article that could be saying, do canines like turquoise? It's pretty much saying the same thing, but I'm using different words. That paper could never be picked up potentially in my search strategy because I only used the words dog and blue. I didn't use the word turquoise and canine. So if I were creating a search strategy, I would begin by listing out every word that could be used for dog, and then I would list out every word that could be used for blue, different shades of blue. There's a lot of different spellings, different variations of words, so I would make an exhaustive search strategy. That way, two things aren't happening. One, you're not missing a lot of papers that could contain data you want, and two, you're not then trolling through lots of papers that definitely don't contain data that you want because you didn't spend enough time on your search strategy. So, search strategy, make it as exhaustive as possible is my advice there. I'm going to talk about deduplication. So when I do my search strategy, I mentioned PubMed. I search many different databases usually. So I'll probably do between 6 to 7. Most of my research is medical research, so I choose scientific databases, but there's loads of different search databases you can use. But yeah, so if you're searching multiple databases with the same search strategy, inevitably you're going to come up with the same papers. There's going to be different papers as well, but you will have a lot of the same papers. So when I check all of my results into EndNote. That's my first tool I use after my database search. When I chuck everything into EndNote, so my first step is to deduplicate those papers. So I go through my whole list, making sure each paper I only have in there once. That way I'm not looking at multiple papers multiple times, because that just takes forever. Once you've deduplicated your library, you have all of your papers, all of your references, and they're only in there once, you get to start the systematic review process, title abstract screening. So what you do is you read the title of each reference, and then you think, okay, does this sound like a paper that will include the data I'm looking for? And if that's a yes, then you can immediately just include that paper. If it's a no, then I would go, or don't know, then I would go and read the abstract. Once you've read the abstract, you should have a decent idea of whether this should be included or excluded. The main point in title abstract screening is to exclude obviously not relevant papers. It's okay to be uncertain about all the papers you're including, but the main point is to exclude papers that you for sure know this is not going to have what I need. There's plenty of opportunities later to cipher through everything that you've included. Once you've done that, you now have a bucket of all the papers that you think could be useful in your systematic review. Once you have that bucket, then you're going to look for all the full texts. Usually when I gather all of my references from, let's just keep using PubMed, they're not going to have all the full texts attached to it. It's usually just going to be a reference, which gives me some, baseline information about the study, title, author, location, date, that kind of thing, and abstract. I don't usually have the paper attached. So the next step would be for me to find all of the PDFs that go with the reference that I've included and attach it. And again, I do this in EndNote. So I attach them all in EndNote, and I know EndNote has a find PDF function. I don't find that it finds everything, usually about a third of papers. I usually end up having to find most of those papers myself through a manual search. Once I've put full text to all of the references that I've included so far, I then do what I called a full text screening, which means I pretty much do the same process as the title abstract screening, but on the full text, and I'm a bit more stringent on my criteria. So I will read the full text of the paper and then decide yes or no, does this actually have the data I want? There's no guesswork here. You're reading the paper, you know it does or it does not. So if it does have the data you want, you can then put this into a new include bucket, and if it does not have the data you want, you can just go ahead and exclude that paper. So now you're left with a new include bucket, and all those papers that are included you want to extract the data out of. So usually when it comes to extraction, the way that I usually like to do things is when I'm doing the full text screening, I will read the paper, then decide if it has the data I'm looking for, and if it does, I'll put it into the inclusion bucket that I've created, and then I will go ahead and just extract that paper right away. It saves me so much more time because I've already read the paper. I might as well just, for the full text screening, I might as well just extract it right there instead of reading the paper, including it, going on to the next one, reading that paper, including it, and then once I have everything in the include, going back to the include bucket, reading the paper and extracting, reading the paper and extracting. So you can do things however you want. There's many different ways that you can do a systematic review. This is just techniques or little tips I've learned from doing it both ways and seeing it's much easier for me just to read the paper, has the data I want, and then I just extract it there, right there and then. So the last step is after I've extracted all of my data, I have it in an Excel spreadsheet, and then I go and clean the data, ready for analysis. I do all my analysis through R, so I end up having to do a little bit of cleaning of the data just to make sure everything I'm extracted is standardized. Usually on the systematic reviews that I've been doing, it's not just me working on it, it's many different people extracting the data. As I said, my systematic reviews that I do are usually quite large. So when there's multiple people doing a systematic review, inevitably people will enter things slightly differently, and even though I go over every variable and tell people exactly how to enter it the way I want it to, there's going to be some variation. I do have some note sections where people can note things about the paper that aren't particularly a variable of information I want to extract, just something they feel that maybe I should know about the paper or the study. So I'll be reading the note sections and applying things. So data cleaning does take a little bit of time. So that is my process for systematic reviews. I will also say that I use different tools to help me through this process as I go along. I've already mentioned EndNote. That's one tool that I use in the beginning stages of my systematic review. As I said, after I do my database search, I put everything into EndNote there. I organize all of my references in EndNote. There are so many different kinds of software, systematic review software out there, reference management softwares out there. You know, there's loads of different things you can use instead of EndNote, and there's a lot of different softwares you can use to actually help you conduct your systematic review. I have decided to use the software. I didn't on my first systematic review, but after that I decided to use Distiller SR, which stands for Distiller Systematic Review. There's a couple other ones that I trialed and used and I didn't really like. Distiller was the one that worked best for me. I can do another video I'm thinking of doing, kind of going over each of the softwares I use and why I chose them and what I use them for and what I like about them and what I don't like about them. Distiller works best for me. I can't say I love it, but it works for what I want, and it really helps me in particular because, as I said, I do have quite a few other people working on my systematic review with me at the same time, so it's very helpful. It really helps you manage everyone, and yeah, I can get more into that later. Anyways, I hope you found this video helpful. I hope you found the tips helpful. I would be happy to go into more detail about the systematic review process if that's something you're interested in. There's so much I can say on this. So yeah, let me know if you guys have any questions in the comments or anything more in detail you'd like me to go over, because I'd be more than happy to just make a quick video about that. Yeah, hope you found it helpful. Thanks, and I hope everyone has a great day. Oh, that's not true because I'm including papers that don't.